So good evening and welcome to tonight's event on behalf of the University of Southampton. Thanks to all the staff, students, alumni and supporters from around the world in joining us this evening. So I'm Mark Cragg, Professor in Experimental Cancer Biology and one of the senior principal investigators based in the centre where I'm speaking to you from tonight. So I'll be your host for the evening and I'm delighted to be joined by two of my colleagues from the centre, Professor Simon Crabb and Lara who'll tell you something about their innovative research they're doing, contributing to new immunology treatments. We are also joined this evening by Dr. Eugen Stulz, who's an Associate Professor of Bioorganic and Materials Chemistry, who joins us from the School of Chemistry to illustrate that these days it's really important to work across borders and traditional boundaries to make more rapid progress. But what really unites us is a desire to understand more about cancer and develop new immunotherapy treatments. So just to start with some definitions. So cancer immunology is what we're doing here. It's the study of how cancer and the immune system interact. And what we're trying to do is elicit immunotherapy, which is using the immune system to treat disease, in this case, cancer. And what we're trying to do is to unpick this really complicated system that we have, this microenvironment that cancers exist in. And so some of you may know, but perhaps many of you won't know, that actually cancers really don't exist on their own. They're not just cancer cells, but they're surrounded by other cells of the body, immune cells, fibroblasts, lots of other things that help dictate how that cancer might respond to a given treatment. And the reason why that's so important to try and understand is because all the time in our body, the immune cells are basically patrolling, looking for cancer cells and trying to destroy them. And they do that in 99.9% of the times that they engage with them. But unfortunately, sometimes cancer cells find ways of escaping the immune system, so they hide from it, or they find ways of preventing destruction. And in that case, that's when people present clinically with cancer, because the cancer has evaded the immune system. And so what we need to do with immunotherapy is re-engage the immune surveillance mechanisms, overcome that immune suppression that the cancers might be evolving so that we can start to destroy the tumours once again. So clearly, this is a complicated process and it needs critical mass to understand. And that's why we entered the centre. So it opened back in April 2018, and it brought together scientists, clinicians, and clinical trials experts that work in cancer research all under the same roof. And the other thing that we've recognised as we've been in the centre is it's really important to work across disciplines and trying to engage with mathematicians, engineers, physicists, different types of scientists to try and solve this particular problem of cancer. And more than that, it's not just working with different scientists across our own institution, but across fields, the faculty, the university and beyond. So definitely working internationally and also with industry. And this slide just gives you an example of the types of things we try to do with immunotherapy. All the things on this slide are molecules or cells that are involved in the immune system that someone at some point has tried to leverage to try and treat cancer a little bit better. And I don't have time to talk through them all today, but those of you that attended the seminar last year will remember we talked a lot about vaccines of some description. So Gareth Griffiths talked about vaccines, and one of our speakers, Professor Simon Crabb, is going to expand on that and show you the progress we've made in some of these DNA and RNA vaccines. And then Eugen is going to talk particularly around DNA, this fascinating molecule that encodes our genomes and our genetic material, and how he's trying to use that for cancer treatments. And then last but not least, Lara is going to tell you about a particular immune cell called a natural killer cell, about how cancer cells try to evade these cells and the kind of things that Lara is trying to do to switch those natural killer cells back on. We've got a diverse array of speakers tonight. So we've got Simon, who's a clinician, Eugen, who is a chemist, and Lara, who's an immunologist. And it's really working across those disciplines that we're able to try and make better progress and quicker progress. And so after you've heard each of them speak, we'll have a Q&A session. So please start to think about questions that you might have. Submit any questions that you might have in the Q&A box on the profile, and we will deal with those once we've done our three speaker sessions. And so now it's my real pleasure to introduce our first speaker, Professor Simon Crabb. So Simon is a professor of experimental cancer therapeutics. He's the clinical director of Southampton Clinical Trials Unit based in the centre, and his research is about trying to develop new therapeutics for cancer and do that through the advent of particularly taking things all the way through into clinical trials. And so I'll hand over now to Simon. Thanks very much.
Thank you, Mark. Good evening, everyone. Thanks for joining us today. It's great to have the opportunity to talk about our work. So as Mark said, I'm Simon Crabb. I'm a medical oncologist. I'm a clinical academic. So I spend about half of my week working in the hospital in Southampton, where I spend most of my time looking after people with prostate cancer and bladder cancer. And I spend the other half working in the clinical trials unit, which is basically the ground floor of that brown building that Mark showed a few slides ago, where we have a large team, over 100 people that are involved in running clinical trials across the UK and indeed abroad. And about half of the clinical trials we do are due to cancer, and particularly looking at experimental cancer treatments and increasingly early cancer detection. And so this evening, I was asked to talk about the Cancer Vaccine Launchpad, which is an innovative approach to how we try to include as many patients as we possibly can through the NHS into cutting-edge clinical trials of personalised cancer vaccines. So the first thing I thought perhaps we should just outline is what a personalised cancer vaccine might actually mean. As people are, I'm sure, aware, we've seen an explosion in the technology available over the last few years for undertaking vaccine development. And perhaps the most significant example of that, of course, was the development of vaccines in a ridiculously short space of time against the COVID virus, which was obviously led to a large degree from the UK. One of the things that allowed us to do was show that it's actually practical and functional to develop mRNA vaccines, and that's particularly important in some of the technology you might want to apply to cancer. And in the years since the COVID pandemic, that's exactly what has happened. The practical aspects of how you might go about making a vaccine that is personalised and specific to someone's cancer is firstly that you need to get a tumour sample, and a lot of these clinical trials are done after a patient has had cancer surgery, because obviously that gives you access to the cancer itself. You then need to centralise that sample. It needs to be processed, and then a process of sequencing that cancer's genetic alterations to understand what it was that actually made that person's cancer tick. And as a result of that, you can predict potential for antigens that the cancer might throw up that might potentially be amenable to targeting through the immune system. And that allows you then to predict how you would design and then produce in real time a personalised vaccine that you hope will be active against that patient's cancer. So that's the theory. And in the last 3 or 4 years at most, this is a slide from a clinical study that was published only 2.5 years ago. We've seen a number of very small scale studies that have suggested that this is actually a practical reality, that we can actually develop a vaccine in real time that will be targeted towards that patient's cancer. This is an example in pancreatic cancer, and as people I'm sure are aware, pancreatic cancer is a difficult disease. We struggle to develop effective treatments for it, and there's a significant unmet need at the moment to improve how we approach this particular type of cancer. This is a really small study. It's only 16 patients. But what the investigators did here was to take patients that had all had cancer surgery. They had their pancreatic cancer removed. The investigators then did exactly what we described on the previous slide. They took samples of these patients' cancer, they centralized them, they read the genes that were mutated and abnormal in each of these patients' cancer, and then they designed and created a vaccine to it. Sent that back to the patient's hospital, and the patients were vaccinated together with some other immunostimulatory drugs, and then followed up over time. What they found was that half of these patients had an immune response to the vaccine that they'd been given. In other words, the immune system seemed to respond to the vaccine in the way that you would expect it to, for example, in response to an infectious disease vaccine, but this time specifically to the characteristics of that patient's cancer. And in the patients where that actually happened, they saw no relapses in the coming 2.5 years after surgery. Whereas in patients where the vaccine didn't appear to do that, so the immune response wasn't generated, those patients tended to have a much less good outcome, and sadly a number of them did relapse and die over the next year or two. So this is a small study, but it suggests that this is something that we genuinely can do, at least in some patients. The other thing that's exciting about this is that the technology ought to be transferable. So it should be possible to do this in other cancers. There's no particular reason why you wouldn't expect to be able to use it in a variety of malignancies. So that has thrown up a number of experimental cancer trials led by a number of large pharmaceutical companies that are going on across the globe, testing this approach of a personalized vaccine to cancer, with the idea of hopefully trying to ultimately improve on cure rates. These are complex studies because to do them, you have to get access to the patient's cancer. It needs to be transferred centrally. You've got to actually make a vaccine to that patient's specific cancer alterations, and then deliver it back to the treating hospital in real time. That's a significant logistical undertaking. And so the UK has approached this with something called the Cancer Vaccine Launchpad. This was developed as a collaboration with a variety of bodies across the UK health system, led by NHS England. And in fact, Peter Johnson, who's one of the academics here and is the chief clinician for NHS England. And we've developed and launched this platform study, the Cancer Vaccine Launchpad, which we've run from the clinical trials unit in the Brown Building from Southampton over the last couple of years. This slide is unnecessarily complicated. I would not try and read all the writing on this, but what it shows is the flow of patients through the Launchpad. So the idea is that this allows patients who are having cancer surgery in ultimately, eventually, we hope every hospital in the UK within the NHS, for patients to be signed up to the Launchpad, to allow their sample to be collected in a safe and controlled manner, and assuming that the patient consents to allow us to do this, that they can then be offered the opportunity to take part in one of these clinical trials where it's taking place at different centres across the UK, and to provide a seamless approach to doing that so that we can recruit and offer these clinical trials to as many patients as possible. We started off doing that with an initial clinical trial that was being run by BioNTech, who people will have heard of from the COVID pandemic approach to COVID vaccines. And this was a clinical trial for colorectal cancer. So these were patients who had bowel cancer, they'd had surgery. The hope would be that they would be cured, but they would be at risk of relapse of their cancer. And so in patients where it was possible to detect circulating DNA from the cancer in that patient's blood, there was the opportunity to enter a clinical trial where a personalized vaccine was made specifically to that patient's cancer, and then they had the opportunity to be vaccinated within a clinical trial. This is still an experimental treatment in the coming weeks and months. But what the vaccines Launchpad allowed us to do was to recruit from patients in centres across the country. So there's something here about the democratisation of access to cutting-edge clinical trials. By their nature, these studies tend to run in big, large, centralised cancer centres We want this to be open to people across the whole of Wessex, for example, for a trial that might be open in Southampton, and indeed across the country. And you can see how we've done that from the diagrams here. From this initial bowel cancer trial, we trebled recruitment to this study in the UK as a result of launching the CVLP. And so we feel that this initial pilot approach has been successful and worked. As a result of that, we've then expanded the launchpad, and we now have 80 sites, in fact, over 80 sites across England now signed up and taking part in the launchpad, which effectively means if you're having cancer surgery in England, you are probably having that done in a centre that is part of the launchpad and can offer you the opportunity to take part in one of these trials. The team are now looking to expand this across the devolved nations, and so we hope over the coming year or so that this will become a true UK-wide approach and effectively available to pretty much everywhere in the NHS. Because of the initial success, we're now expanding. We're putting other clinical trials of other vaccines into this platform. So far, we've added, along with bowel cancer, trials in melanoma and head and neck cancer. And because of the success, we are now going to expand this actually beyond vaccines into other innovative, novel cancer immunotherapy approaches. An example of this are a couple of trials that are about to be put into the launchpad that we will recruit people to across the country, of something called bispecific antibodies. Antibodies are part of your immune system. They're part of what allows your immune system to recognize foreign things like infectious diseases, but you can engineer them. And technology has been developed in recent years that allows the development of antibodies that have what's called a bispecific approach. In other words, they are able to recognize a cancer cell from a marker on the cell surface that is specific to that cancer, but also to latch onto cells from the immune system and essentially act as a bridging molecule to bring the cancer cell and the immune system together, but then also activate the immune system in a way that hopefully stimulates an immune response to that patient's cancer in a way that hopefully is therapeutically beneficial. These are really exciting technologies, but they're also quite complex. And so this seems like a useful opportunity to bring these clinical trials into the CVLP and again, expand and promote recruitment and access to these opportunities throughout the NHS. And in terms of our future ambitions, as you'll gather, we want this to be essentially an NHS-wide approach. We're getting there in terms of the number of centres that we have involved in this. As we've said, the logistics of moving the cancer samples around to get them from the patient's cancer centre to the central laboratory, and then the actual vaccine to go back to the patient and be administered is quite logistically challenging, and we want to improve on the pathways for doing that so that we can streamline this approach. And we think this is part of the way that cancer research should develop in the future. And indeed, as people may have seen today, the government launched its new cancer plan. The CVLP was mentioned as a case study within the documentation for this as an idea of how we develop doing clinical research and clinical studies across the UK, again, to democratise this, to make it open to hopefully everyone, and to speed up recruitment and therefore speed up the science. I think that's my last slide, and so I will hand back to Mark.
Fantastic, Simon. Thanks so much for that. A great update on what's happening in terms of vaccination and also what Southampton is doing to contribute to that endeavour. As someone who works on antibodies, particularly exciting about the bispecific antibodies in combination of that, but we really look forward to hearing about the updates of that perhaps next year or the year after. So now we're going to move gears and we're going to hear from our second speaker tonight, who's Dr. Eugen Stulz. So Eugen is an associate professor of Bioorganic and Materials Chemistry at the University of Southampton. So he's going to talk to us about DNA in cancer therapy and how an interdisciplinary approach can accelerate medical research. Thanks very much, Eugen.
Thank you very much, Mark, for the introduction and particularly for giving me the opportunity to talk here about some of the research that we are doing related to cancer. As Mark said, I'm a chemist. And I think that puts me really in a good position to work on not only on cancer, but medicinal aspects in general, because we can attack the issue at the very bottom of it, on the molecular level. And in that respect, I would like to talk about one particular molecule that you all know very well, or at least I'm sure you have seen pictures of the DNA of this double-stranded helix. So I would like to talk a bit about why it is a fascinating molecule to work with, what opportunities do we have, what we are doing here in Southampton, and a bit also what the future will hold and what are the challenges that we have to resolve. To begin with, just a little bit of history. I'm sure you have all heard of Watson and Crick, but we shouldn't forget that Maurice Wilkins and Rosalind Franklin also have been very important in this pioneering work, 1953, which gave us the structure of DNA and this famous double helical structure. But it's not only the structure that they gave us, but also the function of DNA, because before that we didn't really know what DNA is. Why do we actually have DNA? But now we know very well that it is the bearer of the genetic code, which is then transcribed into messenger RNA, and that then acts as a blueprint in the translation to the proteins, and the proteins do all this fascinating and important chemistry in our body that is needed to keep us alive, basically. This is what is called the central dogma of molecular biology, and every living thing on this planet that we know of works according to this particular scheme. It may be different on another planet, we don't know that. Okay. But it's not only this double helical structure that DNA can adopt, it can actually fold into many different structures. For example, what we call non-B DNA, like G quadruplexes, which are very important in gene regulation. They can fold into aptamers, which work like antibodies. They can recognize proteins or cell receptors. And we can actually design DNA sequences that then fold into 2- or 3-dimensional structures, basically these designer molecules, which brings us DNA nanotechnology. And that opens up a huge space of structure and function simply based on DNA that we can now explore in biology, but particularly also in medicine. But why even talk about DNA? Let's face it, DNA is the source of cancer. Every time a cell divides, it accumulates mutations. And every day we get an estimated 30 trillion of mutations in our body. Now, the majority of those don't do anything or get repaired, and we get away with it. But over time, they start to accumulate, and in particular, if this is in oncogenes, then they may turn a cell into an abnormal cell that then starts to develop into cancer. And if we are particularly unlucky, a single mutation can be enough to trigger that. So traditionally, how we do medicine is we try to target a protein. These are very diverse. It takes more than 10 years, over 100 scientists, over £1 billion to get a drug onto the market, which is then often plagued by side effects. Now, if we can target it on the genetic level, then we deal with one particular molecule. We know how it looks like. We know how it behaves. All we need to know is the sequence. And when we can target that specifically, then we can hijack some of the biochemistry of the cell to get rid of that RNA and with it eliminate the protein in question. And that is what is called the antisense strategy. And based on that and similar technologies, there are drugs on the market. But as of today, none of these are yet available to address cancer. There are still a few issues that we need to overcome, and I will come back to that in a minute. Now, what is different here for me in Southampton? So certainly the opportunities. We have a breadth of knowledge. We have a huge amount of expertise. We have chemistry, biology, engineering, medicine, etc. Now, we're not unique with that. Huh? Every good university, well-equipped university has that. What is different here for me in Southampton is how we're working together. Other places I have been before, scientists are equally excited about research, but usually it's a great idea, but I don't have time now. I don't have resources to do anything about it. Come back later. In Southampton, it's really, hey, that's a great idea. Do you have time next week to talk about it? We should do something. And based on that spirit, I have been able to build up quite a few interdisciplinary collaborations across faculties. And I would like to pick out just one of them to highlight here. That is together with Professor Sian Lim at the Center of Cancer Immunology. You may have heard of her before. She's really pioneering in working with monoclonal antibodies as immunotherapy, in particular anti-CD20 to tackle B-cell lymphomas. And what we are trying to do together is to improve the activity of this antibody. And what we do is we attach DNA to the antibody. And why would we want to do that? Because we can use that then to link it together with other functional designer molecules. So we use chemistries that I just show here, but don't want to bore you too much with it. But what we have designed is this nanostructure out of DNA, which forms a helical bundle, which we can then modify also with hydrophobic molecules. You will see on the next slide why we do that. But you can also see working with DNA really becomes an engineering problem, a designer problem. And the idea is, oops, I've gone one too quick, to have the nanobody or the antibody that is self-targeting, so specific recognition, which then introduces nanopore that can insert into the cell membrane, aid killing of the cells through membrane disruption, and also try to deliver therapeutic DNA into the cancer cell that can then do further action. So what are the challenges? Why are we not on the market with that yet? Biological stability, certainly. You cannot just inject DNA into your bloodstream. We're not meant to have DNA in our blood, so we have a very well-functioning defense mechanism against it. It would be degraded within seconds or minutes at most. We can address that with elaborated chemistry. Off-target effects, meaning that we would target a gene that we don't want, is not always easy, but we can use computing to scan the genome, so to avoid that. Delivery is our major problem because, after all, cancer cells are our own cells. It's not an infection, right? So how do we detect and distinguish a cancer cell from a healthy cell? How does the body that? Because by and large, they show the cell the same surface chemistry. So that's our big challenge to overcome. How do we do that? We need more innovative basic and applied research. Mark already mentioned the interdisciplinary approach. We certainly need to learn from each other. We need to talk to each other. We need teamwork. We do a lot of that in Southampton, but we cannot do everything here because it's just too much in the end. And one thing that Southampton also allowed me to do is really get onto the international stage. And I have been lucky enough to get European funding, a lot of it, for specific programs. One oligomet, which has just come to an end now, and the other one on track based on it, which is actually starting right now, trying to use DNA or oligonucleotide technologies to tackle cancer. And these are some of the people that are involved in the previous and in the current programs across Europe. It's very interdisciplinary. So, for example, Dorota and Annemiek are designing help to design new chemistries to make DNA. Philip is using G quadruplexes to address pancreatic cancer. Carlo is making lab-grown organs so we can avoid animal testing. Christina is upscaling so we can have a lot of material. And Shalini and her team at AstraZeneca help us to bring our systems closer to the market and with it closer to the bedside in order to hopefully have at some point soon a personalized drug against cancer based on oligonucleotide therapy. So I'm very excited about this program. And most of these people actually have been here in Southampton last week so that we can properly start this project. Cross fingers that we have some good success in the near future. So there's a lot of work to be done, which we do, but I hope I could show you that DNA is much more than just genetics or just the genetic code. But once we know the code, in particular the one that we need to address, we can then start designing new molecules, make them through elaborate chemistry, bringing in designs from DNA nanotechnology and together with medicine and all the other disciplines, hopefully get a better cancer diagnosis, but most importantly, a better cancer therapy. Right. That's it for me. Thank you very much for listening, and I'm very happy to take any questions later on in the Q&A sessions. Back to Mark.
Fantastic, Eugen. Thanks so much. A really good insight into how we can leverage DNA in terms of therapeutics, and also how we have to work across borders and across nations to really make rapid progress. So thanks so much for that. So our final speaker for tonight is Lara, who's one of our PhD students who's funded by the Cancer Immunology Fund. So Lara's working on a particular cell type called a natural killer cell, and she's going to tell us about the innovative ways she's trying to make those cells more vigorous in trying to kill cancer cells. Over to you, Lara.
Thank you, Mark, for the introduction, and thank you for the opportunity to speak today. So the immune response is your body's way of fighting off disease. And one of the major arms of the immune response is orchestrated by white blood cells. Now, there are many different types of white blood cells, and this reflects the many different types of diseases and foreign antigens that the body can come into contact with. And as Mark said, the main white blood cell I am interested in is natural killer cells, or NK cells for short. I like to think of NK cells as like the soldiers of your body, as they patrol around the body, usually in the blood, and they're able to rapidly respond to disease or a disease cell, such as a cancer cell. So when NK cells recognize a cancer cell, they release these cytotoxic granules towards the cancer cell. And you can think of these like bullets, as they poke little holes in the cancer cell membrane and enter these holes, and this is what causes the cancer cell to die. Now, I'm also interested in a type of blood cancer called non-Hodgkin lymphoma. This is the most common blood cancer type in adults, and approximately 14,000 new cases are diagnosed in the UK every year. So it's still a disease of quite high burden in the UK. Crucially, the lymph nodes are a really key site in this group of diseases, as quite often within the lymph nodes, a cell of the lymph node goes bad, it becomes malignant. So this is where the lymphoma tumor grows. And we also know that within the lymph nodes, lymphoma cells can receive pro-tumor support signals. Now, these signals keep the lymphoma cells really happy, so they're less likely to die, more likely to grow, and more likely to be resistant to standard therapy such as chemotherapy. But my lab group has also identified that these lymph node associated signals actually upregulate expression of shields at the lymphoma cell surface. And these shields protect the lymphoma cells from the lysis ability of NK cells. And you can see this in my data. So if I treat lymphoma cells with lymph node signals in the lab, this reduces the proportion of these lymphoma cells that can be killed by NK cells. Now, in immunology, we call these shields checkpoints. So cancer cells can express checkpoint ligands that then bind checkpoint receptors on immune cells, such as NK cells. And this interaction effectively switches the immune cell off, puts the brakes on their function, so then they can't lyse the cancer cell. So the cancer cell survives, the tumour grows, and this leads to more severe disease in patients. However, immunologists have devised a way around this through antibody therapy, and we call these antibodies checkpoint blocking antibodies. And what these checkpoint blockers do is they bind either the checkpoint ligand or the checkpoint receptor, so effectively disrupts the interaction of the ligand with the receptor. So this means that the immune cell or the NK cell is not switched off and can therefore lyse the cancer cell. So effectively, this antibody removes the protective shield on the cancer cell and improves immune response against cancer. And again, you can see this in my data. So if I add in an antibody, even in the presence of these lymph node signals, I get a much better killing of these lymphoma cells by NK cells. And we think that adding these checkpoint blocking antibodies into treatment regime for people with lymphoma will more effectively disrupt these shields that are on the lymphoma cells, particularly in this supportive microenvironment of the lymph node. And we hope that this may improve tumour growth control in these patients and therefore lead to better outcomes for these patients. So crucially, this research was published in the British Society of Immunology journal, Immunotherapy Advances. And this is really important as it's really important for us to get our research out there, as this means it can have more impact on both the scientific community, meaning like further laboratory research, and also potentially the clinical community, as it could potentially lead to clinical trials in actual patients. And as Mark indicated in the introduction, my PhD was funded by the Centre for Cancer Immunology Talent Fund. So I could not have done this research and done my publication without generous donations to the university. So I really hope that the university can continue to get donations so that it can continue to fund PhD projects like mine. Thank you very much for listening.
Fantastic, Lara. A really good whistle-stop tour of some of the work you've done as part of your PhD around NK cells. I should just mention, when Lara says trying to get the work out there, she really means that. This is her 10th paper that she's published as a PhD student, which I think is certainly a record in my knowledge anyway. So yeah, well done, Lara. She's done a fantastic job. Her PhD Viva is on Friday, so please all wish her luck. I'm sure there wouldn't be any problem with having 10 papers. So yeah, hope that all goes well. So you've heard from our speakers, our expert speakers. So the plan now is to move to the Q&A element of the talk. So we've had a whole host of questions already come in, so thank you for those. If you'd like to ask any other questions that occur to you subsequently, then please still put them in the Q&A box. We'll get through the questions we can answer in the time we have available, but we will endeavour to respond to everyone offline after tonight. So I'm just going to have a quick look at some of the questions that have come in. And I'm going to basically try to field them across to the expert who I think could answer them best, or who I feel might squirm most by trying to answer that question. So I will start with Lucy Sandy's question, which is a personalised vaccine specific to the person or to the type of cancer? And I think it's only right that, Simon, you probably take that one.
The intention is it's personal to that person's cancer. So yes, not to the person themselves, but to the specific genetic alterations that their cancer happen to have, and that is something that changes on a case-by-case basis. in different cancers. So one person's bowel cancer is genetically different to someone else's bowel cancer in a way that you hope a personalized vaccine will be able to address. That's what we hope is the strength of this approach. I think the data we're seeing at the moment suggests that it definitely can work. What we really need to see in the coming next few years is evidence that you can scale this up and you can do it across a healthcare system. It's all very well doing it in 16 patients in a very clever clinical trial in pancreatic cancer, but you need to be able to show that you can actually do that in a way that would be deliverable within, for example, the NHS. And I think that's part of what we hope we're showing is not just whether this can work, but actually how would you implement it? And that's as much, I think that's as important as this kind of clever vaccine technology behind it.
Great. Thank you, Simon. And so we'll move on to the next question. So following from the pancreatic cancer clinical trial, are there any clues as to why some individuals' immune system responds to the vaccine and others don't? So maybe I'll take that one. The reality is that each of us has a unique immune system. We've all had completely unique experiences. We've encountered different pathogens, different bacteria, different viruses. So all of our immune systems are different. Part of that's genetic, part of that is through experience, part of that is how healthy we are at any one time. So there's a huge number of variables that will determine whether someone responds or doesn't respond to a given vaccine. And I think part of the design and part of the kind of research that goes on that Simon was just alluding to is really understanding how do we get certain groups of people that perhaps traditionally don't respond so well to vaccines to mount better responses. And there's definitely work that's going on at the minute in Southampton to try and understand what are those kind of rules, what are the most important things that determine whether someone is going to respond to a given vaccine or not. I don't know if you wanted to add anything to that, Simon.
Yeah, I think that's absolutely true. And we're going to need to learn an awful lot about how do you predict which approach to vaccination, for example, might work in a particular patient? And it is going to be that balance between the specifics of that person's cancer and their immune system and trying to match those two things up. I think we're on the verge of something really quite exciting, but there's going to be an awful lot to learn about how best to implement this.
Yeah, fantastic. Thank you. So the next question is around genetic mutations. So this is from Jessica East. So the question is, genetic mutations, if these are detected at birth, e.g. by a heel prick, would this help reduce cancer cases later in life? Because it's about genetics. Eugen, do you want to take that one?
I can try. I'm not really very good at genetics, to be honest. I'm not sure. I think if we would detect particular mutations at birth that make us prone towards particular cancers, then hopefully we could use that to develop vaccines early on. I'm only aware of some genetic mutations that can lead to breast cancer that make women much more susceptible or likely to develop breast cancer in future. But apart from that, it's difficult for me to say how far we can go. Maybe Mark, you know more about it.
Yeah, so maybe just to add, so there are some mutations which are inherited, which increase your likelihood of developing cancer at some point in your life. And the BRCA mutations that Eugen was just mentioning in breast cancer are certainly some of those. There's a whole host of mutations that would predispose you to becoming more likely to develop cancer, either in general or a specific type of cancer. So certainly screening at an early age could give you some insight into some of the mutations that may or may not happen. But it's the secondary mutations that happen during your life that generally will dictate whether or not you'll develop cancer, whether it will be particularly aggressive. And it also depends on the rest of your genetic makeup. We kind of alluded to it in one of the other answers, which is around whether someone responds or doesn't respond. And part of that is, what's the makeup of your immune system? Are you immune compromised? Are you immune suppressed? So you might not get a cancer even if you have that particular mutation, because your immune system is very robust and you're controlling it and deleting it all the time. But if you became immunosuppressed, then maybe you would then see the result of that cancer. And the reality is, there's a lot of things that can go wrong. I think Eugen mentioned the numbers. Every time a cell divides, there are multiple mutations that are made. There are corrections that the cell does to prevent those errors and correct those errors. But ultimately, it's a lot of interaction of multiple systems, including the immune system, that then stop the cancer developing. So I think that there's an element of, for certain mutations that are particularly strong drivers of cancer, having vaccinations early on with the kind of suggestion of a heel prick is perhaps, a viable strategy, particularly if we can make vaccinations, genetic vaccinations, cheap enough and deliverable in the same way as we do some of our other vaccinations for infectious viruses. Simon, did you want to add anything to that?
Yeah, no, I'd agree with that. And I think this is the challenge, right? So there are some genetic inherited alterations like the BRCA genes, and there are a handful of others where the risk is so great that yes, this is potentially a viable strategy, but most of the genetic risk that we inherit is still, it increases your risks, but only marginally so. And actually what that then interacts with, as Mark said, is your lifetime exposure to the things that you eat, sunlight, smoking, alcohol, all the other potential risks that some of which you can mitigate and avoid, some of which you can't. So it's about that complex interaction with your genetic makeup, which is, for most people in terms of their cancer, is extremely complex. Your immune system, which is specific to you, and then your exposures that determine your modifiable risk to a degree. So I think it's definitely a direction of travel, and something that we do hope will become more realistic for thinking about actually intervening and for risk prevention for cancer. But I think it is more complex for most people than identifying a specific genetic alteration.
Great, thank you.
Can I add maybe something to that? Yes, to find some inherited genetic mutations would not only affect cancer, but there is a whole range of genetic diseases that are actually issues here. For example, cystic fibrosis, Huntington disease, etc. And it's early days, but in future, I think the CRISPR-Cas technology would probably be very promising in that, because it would allow us to actually repair those genetic mutations. And if we know that early on, we could try and start doing it. There already have been trials and applications of the CRISPR-Cas9 technology to address genetic mutations. But as I said, it's very early days. So perhaps in future, this will be the technology that can be used as soon as we detect a genetic mutation, that we can actually go in and try and correct that. There are, of course, a lot of ethical questions that need to be answered along the way, but I would think this is one of the future ways.
Great, thanks, Eugen. And so we're going to move to a slightly related question, which a couple of people have put in. So Philippa has said, given the NHS's long-term financial difficulties, are personalised cancer vaccines likely to be feasible on a large scale in the future? And you partly addressed this, I think, Simon, but maybe you want to pick up on that.
It's a really important question. These are complex technologies. They will be expensive. However, if you cure someone's cancer, the economic benefit of that, quite aside from the benefit to the patient, can potentially stack up. But I wouldn't take away from the kind of point of the question. And yes, these are extremely complex technologies. I think it has to be part of a kind of wider approach to cancer. Clearly prevention is important. We need to improve on how we actually get people to modify their behaviours such that they don't increase their risk of cancer unnecessarily over their lifetime. And that does come back down to the basic things: diet, exercise, not smoking, all those things that we all kind of know about. We then need to get better at early detection. I mentioned the cancer plan that was launched today. It is absolutely central to the thrust of what we are proposing to do in the coming years through the NHS and nationally, is to try to get better at early detection of cancer, because, as everyone knows, if you catch cancer early, you're much more likely to be able to cure it. So I think those initiatives are coming, and they will be cost effective. If you diagnose cancer earlier, it will be cheaper to treat overall, and that's beneficial to the healthcare system. I think the cancer vaccine story does fit into it. The cost effectiveness of it will be a challenge, but actually, so a lot of the other treatments that we use, and it's much better to be using an expensive but potentially curative treatment than an expensive palliative treatment that is not actually going to cure that patient's cancer. So I think taking anything towards cure as opposed to treatment of established disease is, at least theoretically, going in the right direction. So it's an important point, and it comes back to this idea of implementation and actually what is it of value to a healthcare system. But I don't think we should shy away from these complex, highly personalised treatment options just for that reason. I think we should still pursue them. I think they will have a role in the future.
Great, thanks, Simon. So I'm just going to pick up on a question that kind of relates to the answer that you gave, which was from Chris Christophe, which is how important are diet, exercise, and those kind of things in whether or not people will respond. And I think you kind of alluded to it there. Essentially, if you're fit, you're healthy, exercise, all the things that I think we already know about are all the things that are going to help you respond better to any type of treatment. And certainly there's a suggestion that if your immune fitness is a slightly different thing than physical health and fitness, but they often go hand in hand. So I think it's just generally a holistic approach, as Simon was saying. We need to try and do as many of these things as possible. The second part of the question was around supplements, repurposed drugs in the treatment of cancer. And I think supplements, we just have to be a little bit careful about what they're supplementing. Do we actually need to supplement something? Is something deficient? Then it makes sense to provide a supplement. But repurposed drugs, I think, is a really interesting one. So the biotech companies, the pharmaceutical companies have made huge numbers of drugs over the years that had a particular target, and they didn't work for the particular disease that they were targeting. But actually, they are fantastic drugs. And if with better understanding, we realize that a particular protein, enzyme, or whatever needs to be targeted, then repurposing that particular drug that might have been used in diabetes for cancer is a fantastic opportunity, because often the really expensive part has already been done. All the development's been done. It's been to safety trials in humans. And so then repurposing to another type of cancer, for instance, is a really good option. And I think that's definitely something that we and various kind of cancer charities have kind of advocated, and I think that's a really exciting approach. So there's a nice comment here for Lara. Well done, Lara. Best of luck for Friday.
Thank you.
And I'm going to move on to a question from David Crabb. So are there certain types of cancer that are likely to respond to the vaccine approach, and how do you choose which types of cancer to target with this approach? Simon, do you want to take that?
So I think, first of all, I think one of the genuinely exciting things about the personalised cancer vaccine story is that it should be a transferable technology. A lot of treatments that we develop can be specific to the biology of a particular cancer type. And there's nothing wrong with that. That's great. And we've had a lot of successes. And think of things like hormonal therapy for prostate cancer or breast cancer. Sure, they're not going to translate into other cancers, but they've been highly effective treatments for those particular diseases. At the other end, you have things like chemotherapy, which are useful treatments, but they're essentially a fairly blunt tool to try and help treat patients. It's not a particularly attractive concept in terms of how those drugs work. A personalized vaccine is exactly what it says. It's a personalized treatment exactly for that patient's cancer, and the way that its genetic alterations have evolved through the development of that patient's cancer. But actually, theoretically, there's no reason why it shouldn't be just as effective for a lung cancer as, for example, a pancreatic cancer or a bladder cancer. The technology approach should be the same, and that's really exciting because it means that these clinical trials can effectively be stacked up across different cancer types, as is happening, and we can move things forward across in a much more rapid way than you see in some other drug development examples. So I would hope that would be the case. The more tricky part will be, how do we predict and understand the way that patient's immune system will respond? And that, as Mark said earlier, is probably the more complicated part of this, actually. We know how to make the vaccines, and we know we can test them. How do we actually work out which patient will respond and benefit from this, as opposed to one that may need another approach? And I think we're going to learn an awful lot about that in the next few years, about how best to do that.
Fantastic. Thank you, Simon. So apologies, we're going to have to stop the Q&A there. So I hope we've answered some of your questions. We tried to take a selection of those asking slightly different questions. And as I mentioned, we'll try and respond to everyone as best as we can. So it just remains to make a few closing remarks. So thank you for joining us tonight. So you've heard from just 3 of our researchers and clinicians that are developing different and innovative ways of treating patients to get better, developing new immunotherapies, to develop therapies that can treat patients better. You've heard how we're trying to do that across disciplines. So Eugen, as a chemist, traditionally probably wouldn't have worked with an immunotherapist such as Lara, but we're trying to do more of that. So we've now been in the centre 8 years. Amazingly, it's gone incredibly fast. It's been amazing to be in such a great institute where we have real state-of-the-art facilities, and that's really accelerated our immunotherapy research. So what's next? What's next is more deeper developed understanding of cancer immunology and innovative immunotherapies. And I'm very pleased to start to talk about the fact that that's going to be aided by some exciting new developments at the university. So you may have seen in the news about the new institute we have planned called the Institute for Medical Innovation. So that's going to be a new world-class research institute that's going to be built right next door to the Centre for Cancer Immunology. So the idea there is to try and foster more of this kind of relationship that we have with Eugen, a chemist, to bring clinicians, engineers, researchers, basically all together, access to better equipment, more space, and just increase that interdisciplinarity that we have already, but really trying to build it to the next scale, essentially. So really bringing in experts from completely different fields under a single roof. We're really excited, and we're going to be telling you about that later in the year. But the other thing that I'd just like to say after being here for 8 years is that none of what we do, what we've been able to do, would have been possible without the support of donors. The building was funded through philanthropy. That's why we have the building in the first place. And I know that many of you in the audience have already donated to the centre, and thank you very much for that. If anything you've heard has inspired you, then I hope it has, then I hope you consider making a donation. You can do that by post, by phone, by visiting the website, or you can just send me money in the post. That's also fine. And the details are on the screen. Details are on the screen now. And then just lastly, you'll see a short poll come up on your screen in the next few minutes, and we'd ask if you wouldn't mind to complete that. And that's really just to guide us about the kind of things you'd like to hear, the kind of things maybe you don't want to hear so much about from us when we do our next event. And we really value your opinions on those. For any of you that would like to watch the lecture again, we'll be sharing the footage over the coming days, so please keep an eye on your inbox for details of that. And you'll also find details of other events that we'll be putting on in the future. So I'll just wrap up there. I'll thank the speakers that we had tonight, so Simon, Eugen and Lara, for fantastic talks. Thank you and good night.